So let's get started. So my name's Ron Myers. I'm the Director of Editorial here at JoVE. And today we have a webinar from Dr. Mangalam at the University of Iowa on next generation microbiota 16S gene sequencing. So he published with us back in 2019 on this topic. So he's going to update us today on developments that have been made to the topic in the last couple years, and feel free to ask any questions you may have in the chat or the Q&A, and we will go through and address those at the end. Also, I should let you know that we're recording the webinar today, so we'll send you over a link to the video afterwards for your records. All right, so take it away, Dr. Mangalam.
Thanks, Ron. Thank you so much. And welcome, everyone. I hope if you are not on West Coast, because it must be very early in the morning for you. So wherever you are, welcome. So what I'm going to do is that before I go into my talk is that this is a disclosure. I just want to get away with that. And I'll give you a little background to get into context of why we need to study microbiome. And if you have joined this talk, most probably you have some idea or you have some interest in it. And as you can see that every day in news, you will see one or other study showing importance of microbiota and diseases, because generally it is thought that in a healthy individual or healthy state, we have composition of gut microbiota, which help us with the biological function, keeping us healthy. However, something happened and these microbiota are changed during disease condition. And I'm not going to name diseases, but as you can see from this circle all around it, that lots of diseases, when you look up their microbiota and compare with healthy individuals, they are different. And that's what it is called dysbiosis, which is basically a altered gut microbiota compared to healthy control. So why we want to study microbiota? Because, and I'm going to give it from the angle of health and diseases, but you can apply it to any model you are studying. Basically, you want to perform a study in the patient to find out if microbiome is different. And then the way it is like bedside to bench to bedside, because you want to develop a therapy based on microbiome. So the usual cycle is that, and this is what we also do and lots of microbiome lab do, is that you do a patient-based research where you identify microbiome which are different, then you take it to your basic research, utilize animal model or in vitro tools to determine the role of these, how these bacteria are related to disease or can protect from disease. And then you develop, and your hope is to develop some of the human commensal bacteria or bacterial metabolite as potential biomarker if those bacteria are increased in disease, or therapeutic agent to treat the disease. So as you saw my disclosure that I was able to use this cycle to develop a human bacteria with commensal bacteria, which can be used for disease, and they can, again, so bedside to bench to bedside cycle, so translational potential of these microbiome. And 1 decade ago, when I started performing microbiome studies, it was very complex, or it seems to be. And still, if you are not working with, it seems very complex. So I'll try to break it down to very simple so that you can understand each part of it, because it is very important to understand each step so that you can perform a rigorous study which is reproducible. So this is a basic flow chart for any microbiome study. You have a biospecimen, and it can be anything. It can be fresh tissue, frozen tissue, stool. We have done a skin, nasal, oral swab. And in fact, currently we are doing a project with house dust. So basically any environment you want to study, you can use as a biospecimen. And then you have DNA extraction. I'm not going to go into detail about it because it is already in the JoVE article, which Ron mentioned we did in 2019. Then library preparation and next-gen sequencing and data analysis. So most of the things which are happening, it is in the rigors of the study, how you perform a study, and the data analysis part. This year, we have, and I will come to this, that in next-gen sequencing, we are going to make some big changes in there. So how you do these studies, like I said, the first part is DNA extraction, where you get biospecimen, and again, the details of this technology is already there in the paper, but you have to make sure to use a method where even gram-positive bacteria degrade, their cell wall is broken. So that's why you have to use a bead-based method or enzymatic lysis to get a good coverage of bacteria. Then you have to choose a primer, and when initially microbiome study started, people uses different reasons. So give you a little how these are decided. So to use some 16S rRNA is used for bacterial taxonomic purposes because it has a very conserved region. As you can see on this graph, 1 means highly conserved, and as they go down to y-axis, it is hypervariable. So 16S rRNA region have a high conserved region as well as hypervariable region. And you can design primer in your conserved region because they are not changed across the bacteria. And then you can amplify and use these hypervariable region to classify to a bacterial taxonomy. So we use V3 to V4, and that is the method of choice for lots of lab. So one thing I just want to mention, that I was talking to Illumina and some other company. So what you are going to see is that right now it is limited by, as you can see that this is limited by, right now the technology can do maximum of 300 to 300 pair read, which is 600. So you have to have a region which is less than 500 to overlap them. However, new technology are coming up this year or later because there are lots of things going to happen in next-gen sequencing. And you are going to have a 600 base pair read, so that is 1200, which means basically you can cover the whole, almost the whole region of 16S ribosomal. So then you might get a better resolution of taxonomy. So just wanted to update you on that. So the next step after your DNA extraction is PCR amplification. So initial days when they started, these two steps used to be combined in one. But now we do two-step PCR, where in the first step, you do bacterial specific gene and you have an overhang, which is used in the second step to add the barcode. And basically these barcodes are like giving name to each sample, so you can, which allow multiplexing. So once you have done this thing, then after you run it on your gel, because remember that in microbiome or any big data analysis study, you have to make sure that each step you have done quality control. So here quality control is you want to make sure that there is one major band. So as you can see, bioanalyzer, there is a major band at where we expected between 500 to 600. And in PCR2, we added some extra, so that's why it is a little higher, but still there is a single band in PCR2. So once you get these, because you do PCR1 for individual sample, and then in PCR2, you add barcode, and then you pool all the samples together, and then you clean them. And next part is where you do sequencing. You can use any platform you want. We have used Illumina, and we have lots of success with that. And then come data processing, and where you do reference-based approach or diversity-based approach to analyze your data. And I'm not going to go into detail of this because it is already published. So basically, once you get your sequence, you clean it up. So you remove all the singletons, primer dimers, and then you merge your R1, R2. So two main things are there, and I will come to that, is that, so either you can do operational taxonomic unit, but now preferred method is amplicon sequence variant. The difference between those two is that this will assign to specific to a bacteria instead of doing and binning all the bacteria, which is similar to 97%, and then assigning them to a specific taxonomy. And we will come to that. So once you have data, you can do alpha beta diversity, or you can do machine learning, you can do differential analysis, or you can do metagenome to low resolution, not as good resolution as you do with short-gen metagenomic sequencing. So one thing I want to highlight is that doing 16S rRNA and short-gen metagenomic sequencing, you can ask that if you get more from short-gen metagenomic sequencing, why don't I do that? So each one have its advantage, but tell you one thing is that doing short-gen metagenomic sequencing is very time consuming. It takes 6 months to a year to complete a project, whereas the 16S rRNA, you can complete a whole project within 1 to 2 months if you have good facility. And there are lots of tools, and this is not exhaustive list, but I have said what we have used data to, but this is a preferred method because now it can do amplicon sequence variant. Because basically it means that when it gets the sequence, it will match to the database. And sorry about that. So there are multiple databases, Silva, RDP, Green Genes, or NCBI. And if you want to read about it, it is there in this link, which talk about these different databases. Everyone have their pluses and minuses. We use Silva because it is updated routinely. Green Gene has not been updated for more than a decade. So everyone have their pluses and minuses. And so you can go and look at those articles to see. People also prefer Chime. So advantage of data too is that you can run in your terminal using R, but doing Chime might require more expertise. And we have given the code for data too in our JoVE article, or this article, which we also mentioned in that JoVE article. You can go and see details of how you can do it in your own setting. So first is that you do all these to generate abundance table, which is used for all downstream analysis. And again, there are lots of tools. You can use Microbiome Analyst, or you can use Metagen Assist, or we have developed a tool and which is on GitHub because we found that we didn't have as much control on Metagen Assist and Microbiome Analyst. So this you can run in R in your terminal. It's called MicroVis or Microbiome Visualization. And it was developed by a very talented research scholar in my lab, Soham Ali. And you can use this and you can visualize this data. Only thing you have to have a minimal knowledge of R to do all this. And he had made a good tutorial on that. And then you can also, and if you need, you can always email us because we keep developing these, refining these methods. And you online also, you can find lots of different methods. But you can use any one of them to visualize your data. And one other thing, like I mentioned, you can use functional abundance analysis using PyCrust too. So when I published my JoVE paper, at that time, you have to use a Green Gene database for taxonomic classification so that you can use PyCrust. But Christian Huttenhauer, who have developed these, they have now updated this tool so you can use Silva database and then do a functional abundance analysis. And you can do, and again, you can use R or any, you can perform all these analysis on your own desktop or Mac system. So like I mentioned, and I also do a course on rigor and reproducibility where we talk about like the most important thing is in big data analysis is garbage in, garbage out. So you want to make sure that you take care of everything starting from your sample collection. If you're doing a human study, you want to make sure you have age and gender match as much as possible between your case and control. And you have taken care of diet and lifestyle. If you can do a longitudinal sample or take multiple samples so that because sample between one individual doesn't differ as much. And if then you need to take care of and other thing I want to say is that when you do sample collection, you have to make sure that you use the same method for whole study. You don't change it in between. You cannot have a frozen stool sample and then stool sample in some preservative media, because they will be batch effect. So you want to make sure when you design your study, you follow it once for your study all till you complete it. Then there's a technical variation, the primers, you want to test your primers, like whichever reason, you always use a water control, because there is something called ketone, because kit reagents you are using, they might have some low level bacteria, and you can think that, oh, I found something. So that's they call ketone. So you have to do technical replicate. When you are doing DNA extraction, you have to have a QC for that. You can also do same sample with different barcode. And if you're doing animal studies, there is an effect of cage effect, basically, which maybe two different cages just by random chance have a different microbe. So what we do is that we do cage mixing. And so if you look at lots of our paper, we have detail about that you need to do a cage bedding mix between cages before you start the study to balance the microbiome all across. So once you get your data, it is very important to do a data normalization. And there are multiple techniques. Everything, like I said, there are pluses and minuses, and you can go and look into that, or you can talk to your biostatistician. So remember, one thing I always want to highlight, and lots of paper and grant proposal, that there is a bioinformatic part to it, and that's a statistical part. Bioinformatic part is that all these pipeline which you develop for data normalization or visualization, but what stats to use or what normalization and other, I think a biostatistician will help you with that. So you need both. To give you an example of this study here, you see that you have one sample which has 1,000, another which has 11,000. So basically, if you use this data, you are going to get lots of variability to you normalize it. So rarefying is basically you see when you flatten this curve and you take that point like here, and but in rarefying, you discard lots of data. So people use other where is a log ratio or auto scaling, and I'm not going to go into detail of that. And there's a link to a paper which talk about different normalization methods. So you use those normalization methods for your data, because everybody have a different data. Then other thing is, next is differential analysis. You can do Mann-Whitney, Wilcoxon rank-sum test, but some people don't prefer that because microbiome data is compositional. So then we prefer linear discrimination analysis, effect size or left-C. But there are other methods you can use it for differential abundance analysis. One thing I've always like to highlight, and this is not true from microbiome, but any data you do, is you have to be careful about biological significance versus statistical significance. What I mean by that, if you look at these two yellow one, and you see they both are significant and FDR is also significant. However, if you look at the total count, it seems this might be a random chance that you get 5 to 10 bacteria. So you always have to look at your raw data and do filtering to make sure that you are not using some bacteria which is by random chances in your sample and you think it is important. Then these days you can use lots of machine, because you have heard about artificial intelligence, machine learning. So you can use machine learning tool to find out biomarker, like basically bacteria, which is you can use as a biomarker for linked to positively or negatively linked to a disease. And one other thing is that what we have done is, beside bacterial microbiome, we have recently developed a method for fungal microbiome, or which is called microbiome. The method, it's same. Instead of 16S here, you use ITS region for primer. But after that, everything is same. And other thing is that you use a fungal database, but you can use the same data to a script. Only thing is that your database is different. Instead of bacterial, it is fungal. And one thing which is coming up a lot is that now we have multi-omic data. You have microbiome data, you have metabolome data, you have RNA sequencing data. So you can correlate, you can do lots of correlation analysis like we have done here between fungal and bacteria. So we see in healthy control that, okay, lots of bacteria are depleted, are basically reduced when compared to fungi. However, if you look in multiple sclerosis, there are lots of bacteria. There was a positive correlation between fungi and bacteria. So to summarize all this thing is that we know that microbiome play an important role, and 16S RNA-based metagenomic sequencing can be used to determine significance of microbiota in health and disease. And there are lots of online tools. I have given some link in my talk, but there are lots of them. However, the most important thing is carefully study design, quality control, and appropriate statistical test for meaningful data, because now you have to deposit all the data and you don't want somebody come later and tell you that, oh, you didn't use a proper statistical test or you didn't use a study design. So now reviewers are very careful about all these factors. So like if you're doing a mouse study, there is a legitimate control, because if there is an effect of gene and microbiome, then within case one mouse with gene present and absent should be in the same case to show the effect is because of microbiome, not the case. So you have to make all those appropriate study design and quality control. As I mentioned that for sequencing, it is going to be a very exciting, I will say this decade is going to be very exciting, because we have longer sequencing read, which might allow for improved microbiome profiling. And there are novel machine learning tools. So we are now basically developing lots of model like topic modeling and other. Rachel in the lab is developing, where basically instead of doing an individual bacteria, you see a community, how as a whole it is changed between patient and healthy control or any two different groups. So I think that's all for now. So I'll take any question you have. So I see one in the text, so let me go to that. So this is about somebody, Rishikesh asked that in mother we are doing contigs, how we will fix contig length. So I don't use mother, so I think those kind of question most probably is for your bioinformatic or Pat Sloss. You can ask who developed mother. So like I said, everybody have developed their own tool, and since the people who are like Pat Sloss who has developed this tool, anytime if you have any question, you can ask him and he will respond to your query very fast. So Neil asked question is that how accurate is the artificial intelligence functionally data about microbiome function? So like I said that these are very exciting time, and we still are scratching the surface. As you know that ChatGPT is not even 1 month old, but it is all over the news. So we are still working on it. We can't say that how accurate it is, but like I said that whenever we do any modeling, we test it. So we develop a model and we test it. So far we are excited about it. So watch out for publication from our lab or presentation in different conferences about these data. And we can't tell it because we are still developing those things. So nanopore, so one thing which I was not able to talk about is that, so this is basically because PacBio can do 1,000 bases, but the problem is that you have 10 to 15% error. Illumina we prefer because what happened that error rate is very, so if you take Q30, which is basically 1 out of 1,000 is error rate in Illumina, those when we do 300 to 2. So same with the nanopore is that, so it is depending on what question you are asking. Nanopore is good if you want to do a single bacteria and you want to close the whole sequence. So nanopore might be because you can take it and you can assemble it, because for assembly, long read are better than short read. But for doing amplicon sequencing microbiome studies where you know that hypervariable region V3, V4, like if we are doing it, you are talking about very 10 to 20 nucleotide differences. So in 600, if you have even 10% error, that can tell you, it might tell you that this is a bacteria which might be right. So that's why you want a very low error rate if you're doing a microbiome profiling. If you're doing one bacteria, then nanopore or PacBio might be a good option for you. So another question is that primer bias is still a problem for 16S analysis. So one thing which I think technology has taken, so your primer bias used to be an issue when we used to have just one-step PCR, because you had your barcode as well as primer, adapter, everything in the first step. So now, since PCR1, all the primers are same. So the bias is reduced as compared to the old technology when we used to do one-step PCR. Still, there can be little bias in PCR2, so that's why quality control. So same sample you run with the different barcode and make sure that you are getting the same data. So that's what I said in microbiome studies, quality control is the most important thing to make sure. And then you test different primer. For fungal, when we were doing, we use different primer. Neil again had a question, is that if, for example, is a staph aureus microbiota without cell having same infection potential when compared to other? I'm not sure. Neil, can you elaborate on that? Because I'll go to the next question because I'm not sure what. So how about you elaborate on that question? So Vani had a question, is there any taxonomic classification to classify fungal microbiome similar to bacterial classification? Yes, there is. So that's what, if you go to our +1 paper, which we published last year, so we have used data set there and you can look at detail of that, like which primer to use and which database to use. So Matthias Gross has asked, is that how do you deal with low biomass sample, like how dust we are facing issue with proper control? And another part, so I will come to the first part. So one thing is that whenever you have a low biomass sample, I didn't talk about it, but you increase your PCR cycle. So again, you always have to find a sweet spot that because when you increase, so for normal stool sample, we do 24 cycles per PCR in step 1. But if you have low biomass sample, you can do 28, 30, 32, because for saliva, we do 28, or for fungal, even we have done 28 to 30. So for low biomass, what you do is that you increase your cycle number, but at the same time, now you want a good quality control to make sure. So what you can do is that you can make a mock community as your QC to make sure that you are not getting something which is not there, like some false positive bacteria or fungi, depending on what you are doing. And new technology. So the second part of question is that by new technology, do you mean exactly 600 into 2? So this is something, when I was talking to Illumina, they said, yes, 600 base pair read. And that's what they said, but I'm not sure when they are going to come out with that. So another question is from a user, which is asking, is that what is the scope of real-time analysis targeting a specific organism for 16S rRNA comparison with whether any development? So means I'm assuming you are asking, is that with this data, can we do qPCR? If I'm getting it correctly, is that can we do qPCR for a specific bacteria and compare with microbiome NGS data, which is similar to when you do RNA microarray analysis or RNA expression single sequencing, and then you confirm whatever you find. And I think lots of people use it that if you find some bacteria, you go again and make a specific primer, and you can do qPCR with that. I hope that's what you were asking about that. So yes, you can do, you can design a specific primer and you can check. So lots of lab, what they do is that because they think that 16, because you can do targeted analysis. So lots of lab, what they do is that they do 16S rRNA analysis and they found 20 bacteria. And so they design a primer for those specific bacteria and they do PCR for that. But I think that most probably you spend same amount of money and time when you do 20 qPCR for one sample versus, because I think these days sequencing technology has become cheaper. So it might cost the same for you to do an untargeted analysis. So another question is that, is mother and CHIME used for fungal microbiome generation? We cannot, I'm not sure because I haven't used that. Because we have used our data2 tool, and most probably you can ask CHIME people or other people, you can email them to see, because we have totally shifted to data2 because it gives us more control and we can look at each step. So that's why we use data2. And so, yeah, I cannot answer that question. So again, Rishikesh, those specific question and about the each packages or software, I think it will be better if you talk to them, because if your quality, because one thing is that look at your quality baby in your sequencing read, your Q. So basically you use Q25, but might be that in this sequencing run, you might have to use Q20. So another thing is that when we do data2 analysis. So what we do is that we do quality, we look at how that R1, R2 sequencing done. And then we only select when Q30 or Q2. So if it is, we did 300 and only 260 bases are right. So we will just have that 260. And R2 also, R2 is always not as good as R1. So we will take 220 in there. So 260, 220, we have 480 bases. So I don't know specific because I haven't used CHIME since we shifted to do that. And I know CHIME use data2 plugin, but I haven't used plugin in there. But one thing, if you have a specific question, you can email me directly and I can talk to my lab people, because there are lots of bioinformatics students in the lab and they can help you answer that. So Neil is asking the question is that I have conducted a study where I took 2 different samples from the same stool. There are quite a few differences in microbiota. Is this related to a specimen or analysis? It is very difficult to answer this question, and they should not be. So I will say that it might be technical thing. Then, so if you use the same specimen, when you say I took 2 different samples from the same stool, I'm thinking that the biomass is same. So if you are getting a difference, most probably it might be in the technological step. Either you took more stool from the one specimen versus other. So it is very difficult to point out one thing. So that's what, when we start, we always take same amount. So 200 milligram, it means 200 milligram from each sample. And then when we do, after DNA extraction, we measure DNA using qubit, and then we try to get the same amount of DNA. So you have to do QC at different step. And if you have done that and still you are getting, then you have to look at your kit reagent or other thing, because most probably this is kit related, not analysis related. Anonymous attending, thanks for nice talk. How reliable is Spiker's result based on 16S rRNA sequencing? So again, that like I mentioned, it is a low resolution, but if you have a good 16S rRNA read, you will get, it will point towards some functional, but this is not going to be as good at shotgun metagenomic sequencing. It will give you some answer, and I'm not sure it will answer the question you are looking for. So far with this 300 read, because remember how PyCrust do is that it will take that, okay, if you have this bacteria, most probably this bacteria use these genes. So if that same bacteria have different strain and they have different function, you are not going to pick, because it is going to take a type strain of that particular bacteria and based on what is the functional thing they have. So you will be not able to difference. So if you have a biomass which have a very specific bacterial population, maybe PyCrust will be able to help you with that. However, if you have a biomass where the bacteria different is a strain level of same bacteria, then PyCrust functional analysis will not give you much answer. How can I generate the data which is compatible with Galaxy and Hut Lab? Those are a specific question. I think and like I said, and this you will see that people who develop these things, it is like their baby. So anytime you ask anything, they make sure to reply to those questions. So if you have any specific question, maybe person who have developed that Galaxy software or Christian Hut and Howard Lab, you can email them and I'm sure they will reply. Or one thing we have found is that Google is your best friend, or maybe these days OpenAI is your best friend. You can type in their question and it can tell you that how good or bad that are. Even I have found that, okay, if I have to do this analysis, give me R code, and it can give you R code. Aishwarya, I have a question, is that so how to make contact using forward and reverse primer, and it is possible to obtain similar ATC culture and from environmental. So again, I think once you do, so 16S rRNA is going to tell you this is there at genus level. If you are lucky, it might do at species level. If that genera is not very, so like Prevotella, which has 55 of them, so it will be very difficult to specify which of these species are there. So what we do is that once we find out, okay, these are interest, then we culture these bacteria and pick up single colony and do a Sanger sequencing to find out, okay, what are these strain? If this is matches with ATCC database, or we got something new. Are you facing some problem with MySeq V2 reagent kit Illumina? If so, how are you avoiding it? So far, we didn't have any issue. And one thing I learned, and again, I'm not promoting Illumina, but initially when I started my studies, I used to have a reagent from different vendor. But when you get an issue, every vendor put it on another vendor that, okay, if I use a reagent from vendor X and another kit from vendor Y, another kit from vendor G, then if it doesn't work, X will blame Y, Y will blame G. So what I did is that I went to one vendor, and if I have an issue, then I can get that vendor that, okay, I have problem and I use everything from them. So far we haven't had issue, because initially I had lots of issue when we were setting up in first 2 years. We had some issue, but after that we haven't had an issue in long time with the sequencing. And so how do you choose sequencing method, Ion Torrent, Illumina? So like I said that I contacted other companies, but what happened is that lots of companies are, so Ion Torrent had, what they have done is that they have shifted their focus to clinical side. So they're not interested in, it seems, because I tried to contact them and establish with them, but I never got them to help me establishing this pipeline with that. So I cannot answer them. And most probably the reason I thought is because they are more interested in because they have lots of things going on the clinical side. Patient, they're sampling like bacterial identification on other. So how costly? So there are two questions, which is again asking about cost. So we have developed a microbiome core at University of Iowa. So I can talk about, because when we were developing it, we did lots of cost analysis. So at our institute for inside, if you give a sample toward giving you abundance data table, is we $60 for inside and 70 or 75 for outside people, because there is overhead cost. And so, and this is if you look, we have compared it all over US at least, and we have found that we have a very competitive price. And in fact, we are the most cost effective around there, at least what we know. So coming back to your question, some companies charge $120, some can 80, 90, and 60, depending on how many samples you have and other, but cost will be, I will say, from $60 to $120. How can we determine metabolic profile of a specific bacterial species? So I think this is something, and you can use different companies for untargeted metabolomics, like Metabolon, which is one of the company which have established itself. I heard BGI USA is also offering metabolic analysis. Your core might be there. So lung microbiome, I think people have used like BAL or whole, we have done with bronchial lavage, or if you sacrifice your mice, you can do whole lung, or if you do biopsy. So any type of tissue, you can use it. So any general comment on normalization to or not to for every step from sampling to bioinformatics analysis, don't we lose biological significance? So again, like I said, that because each study will be specific to, because you might be analyzing a big biomass sample like a stool, then it will be different what you use as a normalization versus a low biomass sample like we were talking about dust. So if you want to study house dust or some other dust, and if you are studying that, then normalization method, you will be different. So like I said, you have to ask, it's like Goldilocks system. It cannot be, you cannot do too much or too less. And what you do will be depending on your data set. And most probably, like I said, that a biostatistician will be a good friend of you if you have a specific data set kind of question that what. So remember that people use everything. And one thing we have realized that it is better to talk to a biostatistician before you start your study. So you answer all these questions. If you're doing low biomass, what you use for normalization, which is the best method, and a similar way. And can we get recording? I think, yes, Ron and Smita mentioned that this recording will be available to all the attendee. It will be on YouTube.
Yes, we will send that out shortly after the webinar.
Yeah.
All right. So I think that is it for the questions. So thank you everyone for your excellent questions. And thank you for attending the webinar. Dr. Mangalam, I really appreciate it. Great job. And like we were saying, we'll send out an email later today with a recording of the webinar, and feel free to also reply to the email. If you have any questions about the topic or about JoVE, we can help you with that. All right. Well,
yeah. Thanks, Don, Simta, and everyone who joined in. Thank you so much.
best regards. All right. Take care.